Belnick in the room? No? Okay.
Hello everyone, I am Guylaine Lemer. I come from ophthalmology department of Nantes University Hospital in France. I'm very happy to be here and present the result of EUROPE Period 6 beta phase 1-2 study. The aim of this study is the safety, efficacy of an unilateral subretinal administration of EUROPE Period 6b in patients with retinitis pigmentosa harboring mutation in Period 6 beta gene. Here my conflict. So, the Period 6 beta protein play a key role in a phototransduction cascade. When we have a mutation in Period 6 beta gene, we have a loss of cone of rod photoreceptor first and after in the cone photoreceptor. When patients have Period 6 beta mutation, they have a retinitis pigmentosa. Currently, there is no treatment for this disease. EUROPE Period 6b, also named CTX Period 6b, is a AAV5 gene therapy replacement. We have a Period 6 beta gene under the RK promoter control. Here you can see the final design of the study. It is a single-site, open-label, dose-ranging phase 1-2 study. Patients receive one single subretinal injection in the worst eye. There is 4 cohorts of adult patients. We have 2 levels, a low level first in 2 cohorts and the high level in the 2 other cohorts. Totally, there is 17 patients treated in this study, and between each cohort, a DSMC was held to go to the other cohort. The follow-up period is 5 years, and we have 2 interim analyses at 1 year and at 2 years. Here you have the mind inclusion criteria. Patient might have 18 years with RP, Period 6 beta RP. The BCVA change between the different cohorts. For the fourth cohort, we start with patients we have several diseases. The visual acuity might be below 60 editorial letters for the first cohort. For the ending cohort, for the 3 cohorts, visual acuity must be below 75 letters, and the patient must have conserved visual field at 10 degrees. Here you can see all the clinical or safety assessment who have been done. In blue, the safety assessment. We add laboratory assessment for evaluating the immunological response of the vector shading. In purple, you can see a lot of the efficacy parameters. They are ophthalmologic assessment, but we add a questionnaire patient, a mobility test, and a functional MRI. You can see here the finding of the safety finding of clinical finding. We have noticed a good safety profile with no death or no life-threatening serious adverse effect after this injection. We have noticed 8 SAE in 6 patients, 4 in the low dose group and 2 in the high dose group. 5 SAE related to eyes disorder, choroidititis, retinal detachment, reduced visual acuity, and 2 SAE related to the study treatment, choroidititis, reduced visual acuity. 11.8% of all patients reported at least 1 TAE. This slide shows the multiple consistent signal of efficacy at 1 year across 6 clinical and anatomical criteria, including mobility test, visual test, and FST. All this result is at 1 year post-treatment, 1 year post-injection. All these findings support the extension with a cohort 4 of earlier stage patients on the further clinical step with the preparation of pivotal study. We choose look the natural history to include in the cohort 4 patients with the main inclusion criteria, the age, older than 13 years, no BCVA limit, and patient must be less severely have a severely disease. Visual field must be 20% in all meridian in 5-cat stimulus. Oops, thank you for your attention.
Thank you very much. And while everyone is thinking about questions to all the presenters, if you have any additional questions, do you have a maze, or do you have like a way to measure how they function in real life?
Yes, in this study there is a maze, the aura maze. We test in the different luminosity, and we are just have a look at 1 year, but we have there is few patients, so it's quite difficult to have some statistical data in some patients, because at the beginning the patients are very severely attent, and now they are less attent. So there is a lot of difference in the clinical test and the functional test.
Yeah, because that's what matters to the patients. Yeah, thank you very much. Does anyone have any questions to any of the presenters? Going back as well, if they're still here, they can answer them. And Professor Korobanic and Dr. Brun, you're not in the room? No? Thank you very much.
Thank you.
Then, well, we're going to close the session, give you 10 minutes of your life back. Thank you very much for coming, and have a lovely